So this is chapter 8 in biology, and it deals with Mendelian genetics. And the reason it's titled Mendelian genetics is that the person who is considered the father of modern genetics is Gregor Mendel. Gregor Mendel was a monk in Austria, and he was interested in, he observed how different traits get passed on to offspring in organisms when sexual reproduction takes place, and he set forth some of the principles that we know and believe still hold true about genetics. Our understanding of genetics continues to expand, but the principles that were set forth with minor modifications, the basic principles that were set forth by Gregor Mendel in the mid to late 1800s about genetics, we know still hold true, and they have not been disproven. And so Gregor Mendel is considered the father of modern genetics. Gregor Mendel studied pea plants, and he noticed that there are different characteristics of pea plants. Pea plants can be tall or short. They can have peas that are smooth or wrinkled. And he noticed that the flowers can also be different, and he referred to the flowers as either axial or terminal, so depending on where they came off of the plant. And he noticed that when a tall pea plant was crossed or made to sexually reproduce with a tall pea plant, the offspring were always tall. And when short plants were crossed with short plants, the offspring were always short, and they were never in between. So if you crossed a tall plant with a short plant, you didn't get a medium height plant. If tall plants were crossed with tall, you always got tall, and if short were crossed with short, you always got short. And so he came up with this idea of true breeding. And his idea of true breeding means that certain characteristics are always passed on to the offspring. So he used the terms true bred with respect to that characteristic. So as I said, if a tall plant is bred, the offspring are always tall. That would be an example of true breeding, where the certain characteristics, in this case the tall height, is always passed on to the offspring. So Gregor Mendel came up with some principles of genetics. I'm just going to state them. I don't want you to know them, because we're gonna update them a little bit. So he said that he felt like the traits of an organism are contained in packets of information that he called factors. And he said that each organism has 2 factors. So for each trait, there are 2 factors. And of these 2 factors, each parent contributes 1 factor. And lastly, he said that for every trait, there is a dominant factor. And if that dominant factor is present, then that trait will be expressed in the offspring or in the organism itself. Okay, so these are the basic principles of genetics set forth by Gregor Mendel. And as I said, we've updated them a little bit with regard to terminology, but the basic principles still hold true today. So what we know today is that the information that Gregor Mendel called factors, we now refer to as genes. So the genes contain the information that determines the expression of different characteristics. And we know that for each characteristic, there are 2 genes, so they come in pairs. And if you remember from last module, we said that chromosomes come in homologous pairs. So for humans, we have 23 pairs of chromosomes. So homologous means these 2 are alike, these 2 are alike, those 2 are like each other, those 2 are like each other, etc. So humans would have 23 pairs of chromosomes, and we said that of those 23 pairs, 1 pair determines whether you are male or female. The others determine other traits that you have. So when we say that each of these characteristics is on a pair of genes, so that means that each characteristic comes on a homologous chromosome, a homologous pair of chromosomes, and there would be a spot on each of the chromosomes that is responsible for a certain trait, and that spot is called a gene. So then we use the wording that we use now to express that is an allele. So instead of using the word factor, we use the word allele. So each gene makes up a pair that's called an allele. So each, actually, let's say each so an allele is a pair of genes that's on homologous chromosomes. So So again, if we have a pair of chromosomes that are homologous, they're exact copies, they look exactly alike, but then there would be a spot on that here and here. So this would be one allele, that would be the other allele. And when we put the two alleles together, then we use the term genotype. So a genotype is made up of the two alleles. And we give those alleles a letter designation. And it's pretty arbitrary, but for example, for tall, we might use the letter T. And then for short, we might use the letter lowercase t. So we use upper and lowercase letters to describe these two alleles and the two letter designation. So a genotype might look like this. Okay, so these are all different genotypes for different possibilities of alleles with regard to the letter T that stands for tall. So the two letter set of alleles that is called a genotype. But then we use the word phenotype to describe what the organism looks like with regard to that trait. So this would be the characteristic that is observed in that organism with respect to that gene. Okay, so if we had a genotype of this, then what would be observed from that would be a tall, if let's say it's plants, it would be a tall plant. Okay, if the genotype was this, then the phenotype also would be a tall plant. But if the genotype was this, that would be a short plant. Okay, so the two letter designation is called the genotype. And the observable trait, what you can see, is called the phenotype. Phenotype. Okay? Phenotype. That's really sloppy. Let me do that again. Pheno, it's all one word, but I'm going to have to break it up. Phenotype. Okay? The observable trait. A couple of other terms. If the two letters in the genotype are the same, then we say that that organism is homozygous for that trait. So homozygous means that the two letters of the genotype are the same. So this would be like TT. The two letters are the same, or TT. Okay, that's homozygous. Heterozygous is where the two letters of the genotype are different from each other. So a heterozygous would be big T, little t. Okay, so homozygous and heterozygous, those terms refer to the genotype, and then a couple other terms that we use then are dominant and recessive. So you can a dominant allele and a recessive allele. So a dominant allele is one in which if it is present, it will always determine the phenotype. So a dominant allele determines the phenotype. And a recessive allele a recessive allele determines the phenotype only if there are two of that allele. So, in other words, for a dominant allele, you only need to have one of those alleles in order for that allele to determine the phenotype. Whereas if it's a recessive allele, you have to have two of them to determine the phenotype. So let me give some examples to make that a little more clear. So let's say that so we're going to say that T is tall and lowercase t is short. So those are our two alleles for height. So when we say, we would say this is dominant. And we usually, the capital letter is used to indicate a dominant allele, and the lowercase is used to refer to a recessive allele. Okay, so dominant allele is capital letters, recessive allele is lowercase letters. So what we mean by a dominant allele, so remember for every trait there's going to be two, you're going to have two letters, you're going to have a two-letter genotype. What we mean by a dominant allele is in those two letters, if you just have one uppercase T, then it's going to be tall. So the two alleles, if the two alleles are this, that's going to be a tall plant. Or if the two alleles are this, that also will be tall, because for a dominant allele, you only need one of them in order for that trait to be expressed, in this case, tall. For a recessive allele, you have to have two of them, both of the alleles have to be lowercase in order for that characteristic to be expressed. So in this case, the only way that you can get a short plant would be that both of them are the recessive alleles. Okay, so dominant alleles, you only need one of them. You can have two, but you only need one of them. As long as you have one, that trait will be expressed. For a recessive allele, you have to have two of them in order for that trait to be expressed. Okay, so where do these alleles come from? Well, so we said that every trait has two alleles. And what we know about those alleles is that each parent contributes one of its alleles for that trait. Now, this is for sexual reproduction, because in asexual reproduction, obviously, you're just reproducing with one organism. So each parent contributes one of its alleles. To the offspring. So if we can go back to meiosis, remember that what happened in meiosis was that you ended up with cells that had just half of the genetic information, so that when these come together to form a new offspring, so on this chromosome there's going to be different traits, and each parent will contribute just one of their alleles to the offspring, and then depending on what is contributed by the male and the female, that will determine the genotype for the offspring, and then obviously the genotype determines the phenotype of the offspring. So basically what meiosis does is that it separates the alleles. So you started out with chromosomes looking like this. So you started out with a pair of chromosomes, and what happens you started out with diploid, and then when you go through meiosis, you end up with haploid cells, okay, that have just half of the genetic information. So if this is the male, you end up with these 4 haploid cells, and same thing happens so these are haploid. And then for the female, the same thing happens, except that remember, in the female, you really only end up with one cell that is functional. The other 3 are just called polar bodies, and they're not function, but these are haploid cells also. So then when fertilization takes place, so we'll give these a tail, so they're sperm. When fertilization takes place and one of these sperm fertilizes this egg, then what do you see? One of the alleles comes from the father, one of the alleles comes from the mother, and you're back to having a diploid cell. Okay, so the bottom line is that that meiosis separates these alleles, and that is how the offspring end up getting half of their genetic information from the father and half from the mother. Okay, so there is a technique that is used to be able to predict what the offspring will look like when you cross or mate organisms with different characteristics. And that technique is called a Punnett square. And we're going to start by looking at Punnett squares that deal with only one trait. But they'll get more complicated, and you can look at multiple traits at a time. But we're going to start with just one trait. Okay. So let's say that we are going to cross a homozygous purple plant. And we use the letter X to show that we're going to cross those or mate them. We're going to mate that with a homozygous white plant. And we want to know what will the offspring look like. And the other bit of information that you need to know is that purple is dominant. Okay, purple is dominant. Okay, so that's the information we have. With that information, let's look at what would the two parents look like, and then we'll draw a Punnett square so that we can see what the offspring would look like. Okay, so homozygous purple. So we're going to use the letter P, and this says that purple is dominant. So we're going to use a capital P for purple, and therefore lowercase p would be white. Okay, because we were told that purple is dominant. Okay, so now homozygous purple. Homozygous means that both of the two-letter genotype are the same. So homozygous purple would be big P, big P. And then homozygous white would mean that both letters are lowercase p. Okay, so the two letters, homozygous means that the two letters are going to be the same. So we're going to cross capital P with lowercase p, lowercase p. So to draw a Punnett square, what you do is you draw a square, and we're on the side of that square, we're going to put the genotype of the two organisms. Okay, so here's our square, and so the homozygous purple plant would be capital P, capital P. Okay, so this is homozygous purple. And then we'll put the other plant's genotype on the other side. So this would be homozygous white. Then all you do is, so in these boxes, we're going to we're going to get information about what the offspring would look like. So all you do is you match up. So for this box, it would be a capital P and a little p. This one would be capital P, little p. This would be capital P, lowercase p. And this one would be capital P, lowercase p. Okay, so this tells us what the offspring would look like. And this tells us the potential of the offspring every time a cross takes place. So this says that 100% of the offspring are capital P, lowercase p. Now remember we said that this is dominant, and for a dominant trait, all you have to have is one of those in your genotype, and that trait will be expressed. So this says that 100% of the offspring would be purple. And 100% of the genotypes would look like that. Okay, so that's how we do Punnett squares. Let's do another one. We'll still use P plant. So what if you have a heterozygous purple, and we're going to cross that with a heterozygous purple. Okay, so again, lower uppercase P is dominant, so that's purple. Dominant. And then the lowercase p is used for the recessive allele. Okay, so if it's heterozygous purple, so heterozygous means that the two-letter genotype, the two letters are different. So this would be an uppercase P and a little lowercase p. It's purple because it has the dominant allele here. And then we're going to cross that with another heterozygous purple. Okay, so that this is what the genotypes would look like for each of those flowers. So then we can draw the Punnett square. We make our box. And we're going to put the genotype of one of them on the top and the genotype of the other plant on the bottom. And then we match up the letters. So this would be uppercase P, uppercase P. I didn't draw it. I'm going to make that more clear that that's an uppercase P. Okay. So this would be uppercase P, uppercase P. This would be uppercase P, lowercase p. This would be uppercase P, lowercase p. And lowercase p, lowercase p. Okay, so now we have, these are the different genotype possibilities for the offspring. This one, this one, this one, and this one. And then we can determine what the phenotypes would look like. So first of all, let's do the genotypes. So the genotype possibilities are, here we have an uppercase, uppercase. Then we have, so 1 out of 4 is uppercase, uppercase. Then we have a uppercase P and a lowercase p, and 2 out of 4 are those. And then we have a lowercase p, and 1 out of 4 would be those. So this would be 25%. This would be 50%. And this would be 25%. So in other words, the probability when we cross a heterozygous purple plant with a heterozygous purple plant, the probability is that 25% of the offspring would have this genotype, 25% would have the heterozygous genotype, and 25% would have the homozygous recessive genotype. Okay, so that's the genotype, but then what do they look like? Okay, so this would be purple. These would also be purple because as long as you have one uppercase P, you're going to be purple, and then these would be white. So in terms of the phenotype, in terms of what they actually look like, 75% would be purple and 25% would be white. Okay, so now keep in mind these are probabilities, so they're not always going to come out exactly like that, but if you bred enough of these flowers over a long period of time, generally you'd come pretty close to these percentages, but on each individual breeding you may not get exactly like that. Okay, now let's do a homozygous, a heterozygous purple. And we're going to cross that with a white plant. Okay, so the first thing you have to do is figure out what is the genotype for each type of plant. Well, heterozygous purple. So heterozygous means the two letters are going to be different, and it's purple. So it's going to have the dominant expression, but it's heterozygous. So it would be a capital P, lowercase p, and then white. White has to be homozygous, and it has to be homozygous with the lowercase letter. Okay, so then we draw our Punnett square. And we're going to put this across the top and this across the bottom. It doesn't really matter which one you put where, as long as you get one genotype on one side and one on the top, and then we match these up. So this would be uppercase P, lowercase p, lowercase p, lowercase p, uppercase, lowercase, and lowercase, lowercase. Okay, so then when we look at the genotypes of the offspring, we would say that, so the two possibilities we see here are uppercase p, lowercase p, and lowercase, lowercase. So 50%, 2 out of 4 have this genotype, and then the other 50% have that genotype. So these are heterozygous purple, these are homozygous white. So for phenotype, these would be purple, 50% would be purple, and these would be white because they have both of the recessive alleles, so 50% would be white. Okay. Let me give you another trait where you can see if you can do this. So one trait that is determined by your genetics is whether or not you can roll your tongue. And the ability to roll your tongue is dominant. So it's a dominant trait. That means that if you just have one of the dominant alleles, you will be able to roll your tongue. So let's say we have a heterozygous woman for this trait, and she marries a man who cannot roll his tongue. So the man cannot roll his tongue. Okay, so the first thing we have to do is determine what would their genotypes be. What would the genotypes be for these two people? So the heterozygous woman. Okay, so heterozygous tells us that the two letters in her genotype are going to be different. And let's use the letter R, big uppercase R and lowercase r. Okay, so she's heterozygous and she can roll her tongue, then this would be her genotype. Okay, and if he cannot roll his tongue, so that's the recessive trait. The only way, the only genotype he could have and not be able to roll his tongue would be that. Okay, so these two are going to have children, and we're going to see what are the probability of their children being able to roll their tongue. So the woman will put across the top, uppercase, lowercase, and then the man who cannot roll his tongue would go there. So then we match these up, uppercase, lowercase, 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 uppercase, lowercase, lowercase, lowercase. Okay, so in terms of their genotypes, we would say that, so the different possibilities are uppercase, lowercase, that's 2 out of 4, so 50%. And then lowercase, lowercase, that's also 2 out of 4. So that represents 50% as well. So these people could roll their tongue. So phenotype, 50% can roll their tongue, and 50% are non-tongue rollers. Okay, so does this mean that if this couple has 4 children, that 50% of them will be able to roll their tongue and 50% will not? No, because these are probabilities, and this gets repeated every time they have a child. So it's entirely possible that if they have 4 children, it's entirely possible that all 4 would be that. So it's probabilities, but generally speaking, we could estimate that that 50% of their children would be able to roll and 50% would not. If we had thousands of couples with these genotypes who had children, then when we looked at a large number, probably pretty close to 50% would be tongue rollers and 50% would be not. Okay, so I hope that is that Punnett square is clear to you in terms of being able to look at being able to know what the trait of a male and female is and be able to get a good idea of what the offspring would look like. Okay, I think we will do one more thing today, and that thing is called a pedigree. And what a pedigree allows you to do is that pedigree kind of allows you to work backwards so that you can follow a particular genotype, a particular phenotype through several generations. And using that, you can actually come up with the genotype of that. So I'm going to draw a pedigree up here, and then we'll talk through it. Okay, so let me talk you through this. This is a figure that demonstrates guinea pigs. And a couple things you need to know. So the squares are males and the circles are females. And then the ones that are colored in are black and the others are white. Okay, so this is black. And if they're not colored in, it's white. Okay, so with that information, then what this is it is different generations of guinea pigs getting, and we show which ones get mated with each other, and we see what their offspring would be. So here we have a male and female guinea pig, and this this group mates and this group mates. So we call that the P1 generation. It's like the parent generation. Okay, so let's pay attention first of all to, let's just pay attention to the left side. Okay, so let's just look over here on this left side. So we had a black male mating with a black female, and 3 of their offspring were black, and 1 of their offspring was white. Okay, so now we can think through what their genotypes needed to look like in order for them to be white. Okay, so if we use B is black and lowercase b is white. So lowercase b is a recessive gene. So this guinea pig, in order for this guinea pig to have been white, their genotype had to be this, right? So how is it possible for them to get this genotype? Well, they had to get a lowercase b from each parent, right? And because the male is black and the female is black, they have to have the dominant gene as well. So their genotype would be that. Okay, and so then we can't really predict what these would be because they could be one of these could be B, capital B, or they could be capital B, lowercase b, and still be black. Okay, so they could be this, or they could be this, because they could have gotten, each of them could have gotten a capital B from the parent, but one could have gotten, a capital B from one and a lowercase b from the other. So we don't know what these are, except we don't know what their genotypes are, except that we know that they are black. Okay, what was that? A circle, a square? Okay. Okay, so then let's look at the other side over here. So here we had a white male. So if he's white, we know that his genotype had to be this. And we had a black female, but they have white offspring. So the only way these guys can be white is that they have to be lowercase b, lowercase b. So that means that this he's black, she's black, so she has to have the dominant allele, but she also has to have a recessive allele. That's the only way you could get a lowercase b and a lowercase b together to get those. Now this guy, now we know what his genotype is, because he would get the lowercase b from the dad, and because he's black, he would have to have the uppercase b from the mom. Okay, so now we know these two genotypes, and now those are going to mate, and 3 of them are white. So again, we know that they have to be this, right? And this is black, so we know he has to have the dominant gene, and the only thing he can get from the dad is the recessive gene. So that would be his genotype. Okay. So we can't say what the genotypes of these are by looking at the pedigree, but we can learn a lot about what the parents of the different generations look like. This is the parent generation. Then the next generation, where these mate, this is called the F1 generation, and then these are called the F2 generation. And you could keep going, and pedigrees can be quite extensive, but that's a kind of a simple one. And one time when pedigrees are really helpful is if you have an animal breeder who is breeding multiple generations, and if you have a certain trait that shows up, then you can selectively breed. If that's a desirable trait, you can selectively breed for that trait. But on the other hand, if there's a trait that shows up that is undesirable, then you can make sure that you don't breed animals that might have that trait, so that it doesn't get passed on to the next generation. When I was in graduate school, I worked at the University of Tennessee Veterinary Hospital, and I remember we had one case of so in Dalmatian dogs there can be genetic deafness, and we had a litter of dogs that were brought in which some of them were deaf, and the breeders were wanting to figure out where that deafness originated so that they could eliminate that dog as a breeding partner so that they didn't pass on the deafness to other dogs. So that's a case where pedigrees can be really helpful, is to be able to look at offspring and then work your way back and figure out where that genetic trait might have come from in terms of which of the parents that might have originated with, so that you can could eliminate that trait in future offspring, future matings and future offspring. Okay, I think we're going to stop there. Let me throw out one more thing before we stop. What we have done so far is called monohybrid crosses. And that's where you look at only one trait at a time. But another type of cross is called a dihybrid cross. And that di means two. So that's where you look at two traits at a time. And so you might look at you might have a plant that is heterozygous tall. So this is a tall plant, but it's also homozygous white. So this is white. And then you might cross that with another tall white plant and see what the offspring would look like. So when you look at two traits at once, that is called a dihybrid cross. And I think what we'll do next time is we'll start by doing a dihybrid cross. You can actually look at, it can get really complicated where you can look at even more than two, but we will only look at two traits, and we'll start next time by looking at a dihybrid cross. Okay, thanks. That's the end of part 1 for Module 8.